So in this video, we're going to talk about why genetic engineering is important and how we can make genetically modified organisms. So we need a definition. So we know genetic engineering is the deliberate modification of the characteristics of an organism, maybe a bacterium, for example, by changing its genetic material. And the organism is called a recombinant organism. Now, why do we need it? Well, the first one we just said, we know bacteria can be genetically modified, so we might insert a human gene, for example, into a bacterium in a plasmid, and that human gene can express a human protein. So the bacteria can start to make the human protein, which could be collected and used, for example, as a medication like insulin, or maybe as a vaccine. We could also make genetically modified human cells, and we could then put them back into a patient, and those cells might be able to express the healthy gene to make the healthy protein. It could also be used in germline therapy, whereby, for example, an embryo is genetically modified, whereby we override a disease allele with a healthy allele that's been inserted into the cells of the embryo. So the first step in genetic engineering then is to identify and then isolate the target DNA. So for example, if we had a beneficial gene that we wanted to isolate, maybe the human insulin gene, what we could do is use what's called a gene probe. Now we'd have to isolate the DNA from the nucleus of a cell. We could then denature all of the DNA to separate all the strands. And we could digest the DNA or cut the sugar phosphate backbone with restriction enzymes. We could then add a single-stranded gene probe, maybe with a radioactive or a fluorescent marker that was complementary in sequence to the sequence of interest that we wanted to isolate. So we can see here we've got a single-stranded DNA probe that's labeled with radioactivity. That probe is complementary in sequence to our gene sequence. We add the probe, it binds or anneals, if you like, to the gene sequence of interest. We can then isolate the single-stranded gene of interest, and we can make double-stranded DNA from it. So we can actually isolate the human gene and make a double-stranded human gene using this thing called a gene probe. So, for example, if we had this sequence CAT, AAG, TTT, AGC, so we can see here there's a sequence of codons, and we know each codon specifies a certain amino acid in the polypeptide chain. What would the sequence of the gene probe be used to isolate this gene? Well, we know it has to be complementary to the human gene sequence. So here, the gene probe with the radioactive marker on it might be GTA, TTC, AAA, T, C, and G. So these are complementary codons to the sequence of bases, or the sequence of codons, if you like, in the target gene. Now, another way to isolate the human gene, or to actually synthesize the human gene in this case, is to use messenger RNA. So you can extract messenger RNA from certain cell types, and you can convert that into double-stranded DNA. Now, the double-stranded DNA might be the human gene of interest, and you can put that into, for example, a plasmid, insert the recombinant plasmid into a bacterial cell, as we'll see later, and that bacterium will then make the human protein. So here it would be messenger RNA to double-stranded DNA. Now, one human protein that is a benefit to many people with type 1 diabetes is insulin. So, for example, the pancreas, we know a little bit about, we'll have a little recap, is what we call an endocrine gland. So the pancreas makes this protein hormone called insulin. Now, insulin is actually quite a short polypeptide chain, and it's only 10 amino acids long. And when we've had a meal, the concentration of glucose in our bloodstream is obviously quite high, so we need all the cells of the body to absorb that glucose. So the pancreas makes and secretes insulin, and it gets released into the bloodstream. insulin is it binds to receptors on the cells of all the cells of your body, and that stimulates the cells to make glucose carrier proteins. So the cells of your body start to take in the glucose, and they can then use it in aerobic respiration, and the glucose concentration in the bloodstream returns back to the normal value. So just a little recap about the pancreas then. We know it makes certain digestive enzymes. So for example, pancreatic amylase, pancreatic lipase, and also trypsin. Now trypsin is actually made as an inactive precursor called trypsinogen, and the pancreas cells make another enzyme called enterokinase that activates the trypsinogen into the trypsin form, which is a protease, endopeptidase. So all these enzymes are actually secreted into the lumen of the duodenum, which is where digestion occurs, as well as the ileum, obviously further down, through the pancreatic duct. Now, other cells of the pancreas, called islet cells, we can see them here in blue. They're the cells that actually make and secrete insulin into the bloodstream. So a type 1 diabetic can't make enough insulin because these islet cells are damaged. So if a type 1 diabetic can't make enough insulin, the person can't regulate their blood glucose concentrations, and it remains high in the bloodstream, which can be quite dangerous. So one way of making human insulin genes is to extract the messenger RNA from these islet cells in the pancreas, because we know if they're going to make the insulin protein, the insulin gene is going to be transcribed to a high level to make lots and lots of insulin messenger RNA inside the cells. Now the insulin messenger RNA we know is needed at the ribosomes, and it's translated into the insulin protein. So we can extract these pancreas cells, we can break them open, and we can take out lots and lots of insulin messenger RNA. So this is the technique we might want to use then. So we're going to start with the pancreas cells, we're going to break them open and isolate the messenger RNA. We then purify the messenger RNA, and the next step is to reverse transcribe the messenger RNA into what we call cDNA. So we know transcription normally is where we have a strand of DNA and that's transcribed into messenger RNA. But this enzyme is called reverse transcriptase, so it does the opposite of transcription. Now, obviously the starting template strand will be messenger RNA in this case, so we could actually even add template strand, so messenger RNA. Now, DNA nucleotides in this reaction mix will align next to their complementary base within the mRNA template. And the job of the reverse transcriptase is to link together the DNA nucleotides to form a single strand of complementary DNA, or cDNA, that is complementary to the mRNA. So this is what we'd have after this particular step. We'd have a single strand of template messenger RNA extracted from the cells that normally make lots of this protein. We've made a cDNA complementary to the mRNA. Now we need to get rid of the original mRNA strand, so we can add an enzyme called RNase, and that basically destroys the messenger RNA strand, and it's going to leave the complementary DNA strand. Now again, if we add DNA nucleotides as well as DNA polymerase and a primer, it's possible to then synthesize a complementary strand of DNA against the cDNA strand. So this time in the mix we have DNA nucleotides and we have DNA polymerase to synthesize the complementary strand. So we end up with double-stranded, so we can put here double-stranded DNA. Now one of the benefits of doing this is that we know messenger RNA, or mature messenger RNA, does not have any introns in it, because the introns, the non-coding part of the messenger RNA, is spliced out inside the nucleus after transcription. So the mature mRNA that leaves the nucleus has only coding exon. sequences. So the DNA that we make from the messenger RNA does not have any introns in it. It only has exon coding DNA. So we can put down here only coding, or exon, if you like, DNA. And that's going to be quite beneficial because we're actually going to insert this double-stranded DNA into a plasmid and put the recombinant plasmid into a bacterium, so the bacterium can make our human insulin, for example, for us. So once we've actually made the human gene using this reverse transcription method, we need to insert the human gene into a plasmid to make a recombinant plasmid. So before we can actually do this, we need to isolate the plasmid from bacterial cells. So what is a plasmid? Well, we know it's a small ring of double-stranded DNA found in the cytoplasm of prokaryotic cells, such as the bacteria. So what we can do to isolate plasmids before we start to cut them open is we can actually destabilize the cell walls made of peptidoglycan, so we can kind of damage them and get rid of them. We can add a detergent that's going to dissolve the phospholipid cell membrane, so the phospholipid bilayer of the bacterial cell. And if we dissolve the phospholipid cell membrane, we can then extract the DNA, including the plasmids. Now, once we've isolated our human gene, or even synthesized our human gene from messenger RNA, we need to be able to insert that into our isolated bacterial plasmid. Now, the way we insert the human gene into the bacterial plasmid is by cutting the human gene either side with what we call a restriction enzyme. And if we cut the plasmid with the same restriction enzyme, we generate what are called overhangs or sticky ends. Now, each overhang has a sequence of bases, and if we cut the human gene either side with the same enzyme as we cut the plasmid will have sticky ends or overhangs with complementary base sequences to those to the overhangs of the human gene. So these sticky ends here in blue, they're going to be complementary bases. So we can cut the human gene with the same enzyme as the plasmid to create sticky ends, and they're going to complementary base pair through hydrogen bonds. So if we look at a restriction enzyme in a little bit more detail, then what do they do and how do they do it? Well, one restriction enzyme, for example, is called HindE3. So they've all got these kind of shortened names, HindE3. Now, this restriction enzyme will recognize the sequence AAGCTT. So AAGCTT. And it actually cuts the sugar phosphate backbone at this sequence between these two nucleotides, A and A. So if we look at the sequence at the top of the slide, we can see this enzyme is going to recognize this sequence. So it's got an active site that's complementary in shape to this sequence, AAGCTT, but it catalyzes the breaking of the sugar phosphate backbone between the two A's at that point. So the enzyme recognizes AAGCTT, but it actually cuts the sugar phosphate backbone between the two A's. Now, if you look at the sequence it recognizes on the reverse strand, you should be able to see it's actually the same sequence but going the opposite way. So we know it recognizes AAGCTT, but if you look at the sequence going the other way, it's AAGCTT. It's the same sequence. So this enzyme also recognizes the sequence on the opposite strand. So the enzyme is going to bind to that sequence, so that's the enzyme. And again, it's going to cut the sugar phosphate backbone between the two A nucleotides or adenine nucleotides. And what we end up with once the enzymes catalyzed the reaction is cut DNA. So we can see here on the left-hand side, that's where that DNA strand is cut, which leaves this end here. And on the reverse strand, we can see here it's cut between the two A's again, so it leaves that strand there. So we've got an exposed sequence of bases TCGA, and on the other part of the DNA, AGCT. So these are what we call the sticky ends or the overhangs. Now we know different enzymes, different restriction enzymes, are going to recognize different sequences and cut the DNA at different points. So we've just talked about HindD3. Now another restriction endonuclease is called PST1. And this enzyme, the active site recognizes this sequence of bases. So PST1 here is in green. The enzyme active site is complementary in shape to this sequence of nucleotides, and this time it's going to cut between the nucleotide with a G and an A. Now again, we can see this sequence is CTGCAG, so CTGCAG. But if you look on the reverse strand, go in the opposite direction, it's CTGCAG. So it's the same sequence, just in the opposite direction. So we know PST1, the enzyme, it can recognize this sequence of nucleotides. It binds that specific sequence. And we know it cuts between the A and the G. So PST1 is going to give us sticky ends with these overhangs, and it's going to look like this. So we can see here this strand of DNA has been cut at the G position, and that gives us this overhang here, sticky end. We know on the opposite strand it was also cut at the G, and it gives us that sticky end as well. So here we can say that is the sticky end or the overhang that's going to hydrogen bond with complementary overhangs on a different DNA fragment. Now EcoR5 at the bottom, so we've got EcoR5 down here. This enzyme, it does recognize this sequence, so GAT ATC. So the active site of EcoR5 has a shape that's complementary in shape to the shape of these particular nucleotide sequences, and it's going to recognize GAT, ATC. It cuts between the T and the A nucleotide, but if you look at the sequence on the reverse strand, going the opposite way, it's GAT, so it's the same GAT, ATC, just in the opposite direction. So we know the same enzyme EcoR5 is going to recognize that sequence, it's going to bind to that particular sequence, and it's going to cut between the T and the A. So with EcoR5, when that enzyme cuts double-stranded DNA, obviously it does cut the two strands, but it does not produce any sticky ends. So here we've got kind of blunt-ended DNA. So that's not necessarily going to be useful if you want to insert a human gene into a cut plasmid, because we need the sticky ends to be able to complementary base pair. So let's have a look at our insulin gene, for example. Now this is not the actual sequence of codons that would specify the human insulin protein, but we can use the sequence anyway, just as an example. So we know HindD3 at the top here. We know it recognizes AAGCTT. Now the sequence going in the opposite direction is the same, so AAGCTT. Now we actually call this a palindrome sequence, whereby the sequence in one direction, so AAGCTT, so going from left to right, so maybe 5' to 3', is the same as the sequence going in the opposite direction. So on the opposite strand, going the opposite way, we'd have AAGCTT. So that's a palindrome sequence. Okay, so if we cut the a human DNA fragment to isolate the insulin gene, we'd actually cut it, we can have a quick look at this, between the two A's. So we know this HindD3 enzyme would recognize this sequence here. And we know it also recognized the AAGCTT on the opposite strand because it's the same sequence. And the enzyme would also recognize this sequence down here on the right hand side. And the enzyme would also recognize the same sequence going the opposite way because we know DNA is anti-parallel. So we know one strand goes 5' to 3', and the other strand goes the opposite way, 5' to 3'. Now we know the enzyme is going to cut between the two A's, so it's going to cut the sugar phosphate backbone here, and also here and here. So what we're going to do in a minute is we're going to see that we can isolate the human gene by cutting the DNA here. So we end up with these sticky ends, look. So if we look underneath, that's exactly what we isolate the human insulin gene. Now we've actually cut either side of the gene, and we've got these sticky ends. Now we can then cut the plasmid with the same enzyme, and we can insert the human gene into the plasmid because we know these sticky ends here and the other side on the left hand side here, these sticky ends are going to complementary base pair with those of the cut plasmid. So again, we call these sticky ends. There you go. Now, if we cut the plasmid, so we've got an example of a plasmid here with the same enzyme, so Hindi3 again. We know this enzyme is going to recognize the same sequence as we had before, so AAGCTT. So we can actually draw the enzyme on here. So we know the enzyme is going to recognize that sequence. And again, it recognizes the same sequence in the opposite direction on the other strand because DNA is anti-parallel. We know it's going to cut between the two A's because we can use what we're given up here. There you go. So we're going to open up the DNA plasmid. So this is double-stranded DNA. We're going to end up isolating this bit, and it's going to be separated from the other part, the other end of the circular DNA. So what we end up with is this cut plasmid, and again we've got these sticky ends. So that there is a sticky, what we call an overhang sticky end, as is this one here. Now, if we bring in our human DNA fragment, donor DNA, with the human insulin gene sequence, we know obviously a gene is a sequence of codons, and each codon specifies a certain amino acid that will end up in the polypeptide chain. Now we want to insert our human gene into the plasmid. So if we have a look at the sticky ends for the insulin gene, or either side of the insulin gene, we can see it's AGCT, and the other one is TCGA. So for this one to complementary base pair the sticky end with a sticky end of the plasmid, we know the plasmid must be TCGA. Okay, so we can see that's there, TCGA. So this sticky end here is going to complementary base pair with that overhang. Now here on the right hand side we've got TCGA, so we know that's going to complementary base pair with AGCT. So this bit here fits into that bit. So we're just inserting the human gene into the cut plasmid, and we use the same restriction enzyme to create the same sticky ends, so we've got complementary overhangs. Now this is a summary of what we just said really. So we're going to take a certain restriction enzyme. You don't actually have to use the same enzyme to cut here and here. You can actually use two separate enzymes as long as you isolate the human donor DNA, the human gene, with the same enzymes. So for example, if we cut enzyme 1 here and enzyme 2 here, we could also cut our human DNA out with enzyme 1 here and enzyme 2 here, for example. So we're going to isolate the human gene. We've done that here down below in the middle. We've cut our bacterial plasmid over on the left hand side. The human gene now inserts naturally into the plasmid. It forms complementary base pairs and therefore hydrogen bonds with the complementary exposed base. Now at this point the sugar phosphate backbone of the human insert, so if we just highlight that bit there, has not actually formed a link with the sugar phosphate backbone of the plasmid DNA. So we have to add an additional enzyme that's going to form the link between this donor human DNA and the sugar phosphate backbone of the bacterial plasmid, and that enzyme is called DNA ligase. Now that does not synthesize any DNA at all. It just fills in the bond, the phosphodiester bond between the human insert, which is this one here, and the bacterial plasmid, which is the circular DNA. So this is the bacterial plasmid. Now, why should the same enzyme be used to cut the human DNA and to cut the plasmid? Well, we know it creates exposed sticky ends that are complementary, and therefore it allows the human DNA to insert into the bacterial plasmid DNA by hydrogen bonding. Now we know, just as a recap, if you've got an adenine base, nitrogenous base as part of a nucleotide, and that lies next to a thymine base as part of another DNA nucleotide, here we're going to get two hydrogen bonds. We know adenine is a purine, and we know thymine is a pyrimidine. Now, if we've got a guanine, for example, that's the nitrogenous base of a DNA nucleotide. If that sits next to a cytosine, this time we're not going to have two, we're going to have three hydrogen bonds. Now, again, we know G or guanine is a purine, and we know cytosine is a pyrimidine. Now, you know a purine like adenine and guanine because they have a double ring structure. The thymine and the cytosine pyrimidines, much like uracil in RNA, have a single ring structure. Now, the table on the right hand side, we can see the sequences that are recognized by these different enzymes. So, for example, ALU1 will recognize the sequence AGCT, and it'll actually cut between the A and the G. This one here, TAQ1, recognizes TCGA, and it cuts between the C and the G. And if we actually look at the sequences, the top one, so we'll take this again. If you look at AGCT, if that's a strand going left to right, 5' to 3', if you look at the opposite sequence going the opposite way, so anti-parallel, we can actually see it goes that way, 5' to 3'. So it would actually cut this DNA double-stranded molecule at this point on this strand and that point on the opposite anti-parallel strand. Now, once we've inserted the human gene into the plasmid, we need to be able to insert the recombinant plasmid into a bacterium and then identify which bacteria actually have the recombinant plasmid. So we're going to use marker genes to be able to do this, and they're genes that we find on the plasmid itself as part of the bacterial plasmid. So if we look at the plasmid on the left hand side, we can actually see it's got two antibiotic resistance genes. Now, the one in orange is called ampicillin resistant, and the one in blue is tetracycline resistant. So these genes, they express inside the bacterium a protein, and that protein expressed by this gene will actually allow the bacterium to be resistant to ampicillin. So this protein, it might be an enzyme that can break down the antibiotic so it doesn't kill the bacterium, for example. It might be a protein that blocks the effect of the antibiotic so the bacterium isn't affected, or it stops the antibiotic getting into the cell, for example. So this bacterium with this plasmid will make a protein that gives resistance to ampicillin. It will also make another protein that gives resistance to tetracycline antibiotic. So antibiotic. So it makes another protein that gives resistance to tetracycline in the culture media. Now, if we had a bacterial plasmid, but the human gene, for whatever reason, did not become inserted, then the plasmid would be exactly the same as we've just seen on the left. So we'd have the two antibiotic resistance genes intact. So the DNA sequence of both strands would remain intact. So this bacterium, if it had this plasmid in up here, would make both proteins that we said earlier, so tetracycline resistance and ampicillin resistance. However, it's beneficial for us to insert the human gene into one of the marker genes. And that will allow us to start to select the bacteria that have the recombinant plasmid. So if you look at the picture at the bottom of this slide on the right hand side, this is the recombinant plasmid down here. So we can put recombinant plasmid. And hopefully what you can see is the ampicillin resistant gene, so the orange kind of gene on the left, is intact. The codon sequence is fine, it's not been disrupted. So this plasmid, if it's inside a bacterium, will make the protein that gives resistance to ampicillin. However, the other marker gene, the ampicillin resistance gene, the tetracycline resistance gene in blue, if we look at where the human gene has been inserted, it's actually in the middle of the tetracycline resistance gene. So if we colour the human gene in, let's say, green here, okay, we've cut the bacterial plasmid with a certain enzyme, so it might have been BAMH1, so it's cut in the middle of the tetracycline resistance gene. We've inserted our human gene into this position, so it's disrupted the sequence of codons that would normally make up the tetracycline resistance gene, because we've actually inserted the human gene into this position. So a bacterial cell with this recombinant plasmid, it will make the ampicillin resistant protein, but it will not make the tetracycline resistant protein, because the messenger RNA is not going to be finalized because of the insertion of the human gene, which disrupts the codon sequence of the gene for tetracycline resistance. So we can start to summarize this process by looking at this diagram. Now, we said earlier that we had a human cell, it might be a pancreas cell. Now, we can either isolate the human gene, number one, using a gene probe, or we can actually make the human gene using the messenger RNA isolated from the cell. And that would be reverse transcriptase that we would start to use to make the human gene. Now we know we're going to cut the human gene either side using a certain restriction endonuclease, number 2. And that's going to leave what we call the sticky ends, number 4. Now we know there's a sequence of bases that's going to be complementary to the plasmid that's been cut with the same restriction enzyme. So we know we can isolate plasmids from a bacterial cell. We can degrade the peptidoglycan cell wall of the bacterial cell. We can then dissolve the cell membrane using certain chemicals that will kind of get rid of the phospholipid bilayer. We extract the plasmid. We cut the plasmid with the same restriction enzyme as we did the human gene. We insert the cut donor DNA, the human gene, into the cut plasmid. The complementary bases of the sticky ends hydrogen bond. We then add DNA ligase, which joins together the sugar phosphate backbone of the human gene. So this is the human gene with the plasmid. So this is the plasmid. And that gives us our recombinant DNA. Now we need to get this recombinant DNA into bacterial cells for the bacteria to start expressing the human gene. Now there's several ways that you can do this. One is to give the bacterial cells in the presence of the recombinant plasmid an electrical shock. And that opens pores within the phospholipid bilayer, the cell membrane, to allow the recombinant plasmid to go into the cell. We can also use what's called heat shock. Now we know a short heat shock provides the phospholipid bilayer with more kinetic energy, so the phospholipids can move about more, which creates small gaps in the phospholipid bilayer cell membrane. So again, the recombinant plasmid could go in to the bacterial cell. A third way is to add calcium chloride to the bacterial cell, and again that creates little pores in the cell membrane to allow the recombinant plasmid to go into our bacterial cell. Now once we've got our recombinant plasmid, such as this one here on the top right, because we've got our human gene insert there, and we've put the recombinant plasmids into bacterial cells, we need to be able to select the bacterial cells that have taken up the recombinant plasmid. So we can do this in a couple of ways. The first one would be to plate all the bacteria on an agar plate. So agar, we know, is a type of nutrient jelly, and it has all the molecules that bacteria need to divide and increase in numbers. So it would contain, for example, glucose, amino acids, nitrates, potassium, sodium, phosphates, etc. So this is an agar plate. Now this original one here does not have in the middle any antibiotics added to it. So any bacteria with the regular plasmid with no insert, and any bacteria with the recombinant plasmid over here on the right hand side with the insert will grow because there's no antibiotics. Now what we can do is, once we've got the colonies on this agar plate, so we know one bacterial cell, when it's plated onto an agar dish, if that plate is incubated at 30 degrees C in an incubator for a couple of days, one bacterial cell will form a visible colony, which are what these circles are here. So we've actually got 6 visible colonies on the agar plate. Now it's possible to then transfer some of these bacteria from each colony onto fresh agar plates. Now this time, if we have an agar plate with ampicillin, so this agar plate, ampicillin here, is agar with ampicillin antibiotic in it. Now we can see that the bacteria that's been transferred over, bacteria from colony 1, from colony 2, colony 3 and colony 6 will grow on ampicillin. So you can see them here. If I go over in red, you can see they're at the same position. So we've copied this position of colonies onto the new agar plate. This time it's got ampicillin in the media, and these colonies grow. So we know these could either be bacteria with a regular plasmid, such as this one at the top, because that's got the ampicillin resistant gene. They could also be recombinant bacteria, because we know the ampicillin resistance gene is also intact here in this recombinant plasmid. But if we copy the colonies of bacteria from the original plate over onto a fresh agar plate with tetracycline, what we see this time is the colonies of bacteria that grow are colony 1 and 3. So I'll just circle colony 1 and colony 3. So if they grow on tetracycline, we know that these bacteria must have a tetracycline resistant gene. So these bacteria must have the regular plasmid without the human gene. Now that allows us to work out which bacteria actually have the recombinant plasmid. So we can see at the top right the human gene, if we just do it in green, it's probably a bit better colour here. Well, we know bacteria just with the recombinant plasmid, they will grow on ampicillin, but they will not grow on tetracycline. So any colonies that have grown on ampicillin but not tetracycline will contain the recombinant plasmid. So we can see colonies 2 and colony 6. So colony 2 is made from bacteria with a recombinant plasmid, and colony 6 is made also from bacteria with the same recombinant plasmid, because they grow on ampicillin, but they do not grow on tetracycline, because the tetracycline resistance gene has actually been, if we look at this at the top right hand side, disrupted by insertion of the human gene. Now, some of the pros or some of the benefits of using bacteria for genetic engineering. Well, as we've just kind of shown, we know we can insert human genes, beneficial human genes, healthy human genes into a plasmid. The bacteria will then start to make the human protein. So we can say the human protein. And we can use this possibly in medicine. Now, one benefit of using the human protein as a medicine is that when you inject it into a human, it's not seen as foreign, because healthy people should really have the human protein in their blood. So when you inject the human protein into a diabetic patient, they're not going to mount an immune response to this medication. Now, we said earlier that these recombinant bacteria can also be used to produce vaccines. So imagine you had part of a gene that made a certain polypeptide that was maybe a viral protein. Now, because it's not the whole virus, it's only a part of the virus, it's only one of the viral proteins, we could inject that into people as a vaccine. Now, the whole point of a vaccine is that it allows the person with the injection to elicit an immune response against this particular antigen without the vaccine causing infection in the person, because it's not a full viral particle, it's only a fraction or a part of the protein of the virus. Now, another benefit is to use these bacteria or any genetically modified organism to detect environmental hazards or environmental change, such as the amount of carbon dioxide in the atmosphere, for example. Recombinant organisms can also be grown, things like plants, so we can genetically modify plants. Here we can have, for example, a bacterium that can infect plant tissue, and it can inject the recombinant plasmid into the plant cells, which then start to express the desired gene. So we could make plants GMO organisms. Now, these might be resistant to things like certain pests. They might be more hardy, and they might be resistant to things like frost, so they grow better. They might grow bigger. They might grow more succulent fruits. The fruits might be brighter in colour, for example. So there's a number of benefits. Now, the disadvantages or the cons of using genetic engineering then. Well, we know if we're going to create recombinant plasmids with antibiotic resistance markers or genes, you have to be very, very careful, because you don't want these recombinant plasmids to get into the environment, or the bacteria that contain them to get into the environment, because then we could have problems with the recombinant plasmid with the antibiotic resistance marker getting into pathogenic strains of bacteria, which would then be resistant to any antibiotic treatment. Now, another problem with genetic engineering is that if we put human genes into other organisms, we don't necessarily know if these human genes or human nucleic acid contain oncogenes, because we know human genes can actually be mutated and they can become cancerous, which is what an oncogene is. Now, also, if we insert a human gene into another organism, the cell of another organism or cells, we don't necessarily know where that human gene will incorporate into the chromosome of those cells. So if we put in a human gene into plant cells, we don't necessarily know where the human gene is going to incorporate, and it could actually disrupt the normal functioning of other healthy genes, for example, in the plant nucleus. So we could have a disruptive effect by inserting a human gene into a part of a chromosome that could disrupt the codon sequence of other normal healthy genes. Now, very quickly, we're going to do gene therapy. So if we were to introduce a normal healthy gene into the cells of a person that had a defective allele, that might restore the health of these cells, because the human gene that's inserted into these cells could make or express the healthy protein. Now, it's called somatic cell therapy or somatic cell gene therapy because it's inserting the healthy gene into somatic or body cells. So even if the human gene, the healthy human gene, gets inserted into these cells, into maybe a chromosome inside the cell, and it expresses that protein, the alteration is not going to be passed on to the offspring of that individual, because the healthy gene's only been put in the body or somatic cells. Now, how you would do this then? Well, you would isolate some cells from the affected or diseased region, and we'd use a vector. So we might have a virus that's been attenuated. So we might take out the nucleic acid of the virus. We might modify it by inserting, for example, here, the human gene into part of the virus nucleic acid. We then put the recombinant DNA back into a virus or a weakened virus, and we allow the virus to infect these isolated human cells. Now, we know some viruses are called lysogenic, so lysogenic viruses, and they allow their nucleic acid to be inserted into a chromosome inside the nucleus of the host cell. So you can actually get insertion of the human gene, which is this bit in blue, into a chromosome inside the nucleus of that cell. Now, as long as that gene does not insert into another normal healthy gene, the healthy protein is likely to be transcribed, the healthy gene is likely to be transcribed, and therefore the mRNA is going to be translated and the healthy protein is going to be made. So we can then put back these recombinant cells that have been genetically modified into the individual, and they're going to express the healthy gene. But because the genetic alteration is not in the germline cells that makes the gametes, okay, so the genetic alteration is in somatic cells, and it's not in the germline cells, this alteration is not going to be passed on to the offspring. Now, germline therapy is slightly different because by introducing the corrective gene into an embryo, we know that embryo is going to continue to divide many times, and it's going to form the fetus. So all the cells generated from that embryo when it divides will have the normal healthy gene that's been inserted. So all the cells of the individual that form from this embryo will have the healthy gene inserted into their chromosome, and they'll make the healthy protein as long as that gene is being expressed. And that includes the germline cells that form within that individual. And we know germline cells are the ones that are used to make the gametes through gametogenesis. So an individual that forms from an embryo that's been genetically modified will have the correct healthy gene in the cells that are used to make the gametes. Therefore, the gametes that are produced by this individual will also have the corrective gene or the healthy gene, and therefore germline therapy allows not only an individual to have the healthy gene, but the offspring of that individual to potentially have the healthy gene. So the offspring also may have the healthy normal sequence allele. So this is a summary. So germline therapy, we might take an embryo with a defective gene. We can genetically modify it to insert the healthy copy of the gene into the nucleus. We then implant, or we'd actually use IVF to allow the embryo to develop and divide further. We then implant it into the uterus of a female. The embryo would then develop and divide into a fetus. The fetus is born, And that individual that forms from the recombinant embryo would have the corrective gene in all its cells and therefore make the healthy protein. The modified gene is going to be passed on to the offspring from this person because his gametes or her gametes will contain the healthy gene. Now, somatic gene therapy we know is a bit different. So what we can do is we can take human body cells, or we can actually take them from other organisms sometimes. We're going to genetically modify them and then insert them back into the patient once they've been changed. So genetically modified or changed, and they go back into the patient. Now the modified genes will obviously be expressed, so it allows the tissue to become healthy, possibly in that person, but we know the modified gene is not going to be produced in the gametes of that person because they are not, the germline cells do not have the modified or changed gene. So they do not have this modified gene.